Thank you for kind introduction. Good morning, ladies and gentlemen. I'm Hyoung Jin Kang from Seoul National University, South Korea, not North, I'm from South. Yeah. And first, thanks to Miltenyi Biotech for inviting me to this wonderful summit. And this time I'll talk about the development of investigator-initiated CAR T trial for ALL in Korea with the help of Miltenyi. As you know, CAR T targeted for CD19 from UPenn showed great success. Emily, who was a refractory ALL patient, can be cured. This result was published in NEJM, and Novartis made the first drug of CAR T, as you know. In this aspect, CD19 CAR T is a wonderful model in aspect of humanity, science, and industry. With this success, Tisa-cel became a drug with high price. Tisa-cel introduced in clinical practice in 2017, but not in Korea at the time. So I found a way to treat patients in Korea, and fortunately I found Miltenyi prodigy can be used for the closed automated CAR T manufacture, which was very easy for our institute to make point-of-care CAR T. I started discussion with the Miltenyi Biotech colleague, and finally they agreed to support CAR T clinical trial in Korea. To prepare CAR T production in our hospital, 3 persons, including me, got Miltenyi prodigy training course in 2018, and I met Stefan at the time, as in this photo, and concluded clinical trial research support agreement in 2019. With the government fund for Korea research-driven hospital project, we prepared the IND filing for CAR T clinical trial and start the discussion with our MFDS, Korean FDA, about CAR T clinical study first time in Korea, because before that there's no clinical trial for CAR T in Korea yet. And our one was the first review for the CAR T trial in Korea by FDA, and as our FDA are very conservative, we should make full CMC package and IND dossier like company-driven study. We developed the production system, QA/QC system in hospital, performed GMP production, validation, and preclinical animal study, and got approval of investigator-initiated CAR T clinical trial first time in Korea. Make history of some kind of CAR T in the point of care. This is the overview of Korean cell and gene therapy regulatory framework. There was an act that regulated cell and gene therapy in Korea, which was enacted in 2020. And this act is composed of 2 chapters. One for advanced regenerative medicine, ARM, or for IIT and clinical research, which was governed by Ministry of Health. And the other chapter is for advanced biopharmaceuticals, ABP, for SIT, and commercial approval, which is governed by Korean FDA, named MFDS. But for high-risk clinical research such as CAR T, the regulation is somewhat complex. First approved by ARM, by Ministry of Health, and then approved again by FDA. There's 2 steps. As our one CD19 CAR T was first in Korea, we prepare IND document according to the new regulations. And I will talk of some specific preclinical study that we prepared for CAR T, CD19 CAR T IIT, in addition to the Miltenyi IB. As mentioned in the label of Tisa-cel, CAR T is different from previous medication, so classic toxicology study would not be applicable. It's very amazing that Novartis done that this kind of things, that they persuaded the US FDA and become a drug. As you know, the leukemia occurred for gene-modified cell in clinical cell and gene therapy for SCID, severe combined immune deficiency. So there was a concern about oncogenesis by gene therapy, and you know that there was a certain lymphoma occurred in the CAR T or BCMA, but the reason was not revealed yet. And also our experience, all the experience of cell and gene therapy for chronic lymphomatous disease, some kind of immune deficiency, showed that there was some modified gene located in your oncogene existed for a long time. This cell was exist. And Novartis performed insertional site analysis, and Korea FDA also request this kind of data. So we developed a new integration analysis method named distinct sequencing and perform analysis about CAR T made in our hospital and showed that there is no preferential insertion site. This is the NGS-based analysis and with nested PCR, and this result was recently published. As US FDA mentioned, there's no good animal model for safety for CAR T. Our FDA requested the full preclinical animal study, toxicology, effect, and distribution. So we made the preclinical leukemia xenograft mouse model for GLP level and performed the CAR T, see the effect, the toxicity, and distribution preclinical study, also published last year. It might be the first for the CD19 CAR T. And then we made the point-of-care CAR T production system with the qualification according to our guideline, and paved the way for CAR T production using ProDD in the Korea with the help of Miltenyi. Then we successfully treated the first patients in 2022. This is the first one, and everybody has a mask because there's COVID-19. And I'm not there because I was in COVID-19. And in 2022, 5 patients received the CD19 CAR T made at our hospital, and among them, unfortunately, 2 CD19 negative relapse occurred. B-cell aplasia occurred in all patients means that our CAR T worked very well. Fortunately, Tisa-cel, the Kymriah reimbursement in Korea in 2022, and we plan to close our study. But unfortunately, the reimbursement indication was similar to the inclusion criteria of Eliana study, which excluded patients with hematologic CR. So they are not covered by insurance who relapsed in extramedullary or isolated relapse CNS or MRD relapse or CR after chemo. They are not covered. According to the real-world experience, as you know, patients with extramedullary relapse also showed promising outcome compared with the patient with bone marrow relapse. And among patients who received our CD19 CAR T, one had extramedullary relapse, and as you see in this mass near the heart, this is extramedullary relapse. Teenage girls, and this mass disappeared after the CAR T treatment. So we continue to open our trial for patients who are indicated for CD19 CAR T but not covered by our insurance. Until now, we treated 7 patients. Among them, 5 patients are alive, and 2 patients received the transplant after CAR T. 1 lost B-cell aplasia, and 1 received etoposide to treat CRS, HLH. This slide is the result of last patient, 7 years old, leukemia relapsed as a mass, and he gave chemo multiple times and finally relapsed in mass and CNS, not bone marrow. And he received our CAR T and also showed CR and get transplanted now and be very well. As our CAR T IIT trial was the first IIT in Korea funded by our government, our work was chosen as one of the top 10 scientific news in Korea because we are somewhat behind in this field. And our Ministry of Health and Welfare praised my work as their big achievement. And it's Korean, sorry, but I will explain. And I received some kind of prize over the Ministry of Health to do this kind of things. I saw, thanks to Miltenyi, that I cannot praise owing to you. Yeah. And we can give CD19 CAR T we think optimal. It means that we can extend the indications. And I'll talk of the next step after our CAR T IIT. According to the follow-up data of Eliana for the Kymriah ALS study, the long-term outcome was not so satisfactory. The event-free survival only 36%, even in pediatric ALL. And the relapse occurred in about more than half of patients, half with CD19 positive and half with CD9 negative, and some with lineage switch to myeloid cells with very poor outcomes. So to improve the outcome of CAR T effect, post-CAR T consolidative transplantation is indicated in some patients who are urgently needed. It means that CAR T and transplant help each other to improve the survival. According to the kind of relapse phenotype is associated, like the many treatment before CAR T or big burden, there's a tendency of a CD19 negative relapse, and young age and disease, high disease, CD19 positive relapse and high disease burden and Boolean failure previous CAR T, there's CD19 negative relapse and some MLL rearrangement, there's some lineage switch. According to this result, there's some guideline published that in some patients, transplantation is necessary. And also Chinese group showed beautiful data with consolidative transplantation after CAR T treatment. So, as you know, patients with MRD, minimal residual disease positivity, especially with NGS-based. Recently in Korea, discovered by insurance, and all patients perform NGS-based MRD in ALL. And before this transplantation, showed a high rate of relapse. It is meant that NGS MRD CR is necessary before transplantation to get good result. So I think with high risk patients, such as a high disease burden and tendency to relapse after CAR T, or Boolean previous non-responder, or who have lost B-cell aplasia very soon, within 6 months or within 1 year, and the patients who receive chemotherapy to control CRS like HLH mass. In this indication, we transplant the patient now. And to improve outcome of CAR T, post-CAR, the new indication is necessary, right? We should broaden and we should treat in earlier line, like lymphoma study. As you know, there's some success by Yescarta and some brain and unfortunately in Kymriah. In the DLBCL, there's success just a second line and approved. And this kind of effort is ongoing in pediatric field. And as you know, the disease burden is associated with the trial outcome. High disease burden means that bone marrow blast more than 5% before CAR T infusion. The relapse rate is very high, and the survival is very poor. And interestingly, B-cell is necessary to improve the survival. It means that antigen is necessary to proliferation of CAR T. So considering this kind of result, patient with a low disease burden with enough CD19 cell might be the best indication for CD19 CAR T. Effect of CAR T might be best for patient with a hematologic CR and the B-cell recovery. And in this aspect, patient indicated for transplantation with hematologic CR might be the best indication for CD19 CAR T. Also, allogeneic transplant, which was a curative option for very high-risk ALL, resulting in long-term toxicity such as permanent infertility in almost all pediatric patients, and some kind of lung fibrosis which need a lung transplantation, and permanent alopecia, surely very troublesome for teenagers. To improve outcome of CD19 CAR T and to avoid this kind of transplant and toxicity, clinical trial of CD19 CAR T is ongoing for pediatric ALL with MRD positive after consolidation chemotherapy, means very high risk of relapse. This is performed by COG group and Novartis, and unfortunately not in Korea. But fortunately, we got some big fund In Korea, past chairman Goniri from Samsung company donated a big fund when he passed away in 2021 to help pediatric cancer and rare disease patients across our nation. With this fund, we can start a nationwide multi-center clinical trial for CD19 CAR T for pediatric patients indicated to allotransplant to improve the survival and to even avoid the transplant in 55-year patient. And I hope to present the result next time. And to improve the outcome of CAR T for ALL, the novel CAR T for refractory relapsed ALL is necessary. Like you know that there is a relapse of CD19 negative, and CD19 22 might be a very good option in like the bispecific or co-administration, bicistronic, and tandem. There's many ways to perform CD19 22. And recently, very beautiful data was published by Chinese group by the sequential 19 and 20, 19 CR, and then they give 22 and showed very good survival. And we can find the new antigen such as RR1, which is recently started in ALL trial in pediatric field as antibody. But until now, the CAR T shows not so satisfactory. But some improvement might be given a better outcome. And there is many next generation molecular platform for CAR T under development to improve efficacy and to reduce toxicity, as you know. And one other option is made CAR T with good T-cell. In adult lymphoma study, DNA methylation signature of harvested T-cell, not before CAR T production, has some associated some survival tendency with their methylation pattern, and they named it as epic CAR T. I don't know what the meaning, but they said it. And we analyze again with this result, and the epic CAR T signature of harvested T cell predominantly reflect the naive T cell subset. Means that if we can make CAR T with naive T cell, we might have a better outcome, which should be proved in future. And another big problem is cost and time, as you know. To make CAR T, you know, the leukophoresis, activation, vector transduction, proliferation, and harvest and infusion. Has many process, very complex and need very QC. So compared with the current drug with one production and one QC for many patients, current autologous CAR T is only indicated for one production, one QC for one patient. So there is no chance to reduce the cost with this method. So as you know, financial toxicity is a very big problem. Every nation, everybody, every patient can endure this kind of very expensive new high-tech treatment. There are some way to make cheap CAR T, like the manufacturer innovation. Everybody like these things, and you know that this machine and for this, I know that this is very talented child who has a big power in future, like Mozart. And this may substitute the conventional manufacture, and surely I can reduce the many paper and many work with this automated color system. And manufacturing innovation such as one-day CAR T already published and in the way of a trial, as you know. And some researchers use non-viral vectors such as piggyback for CAR T production, but recently I heard that there is some lymphoma occurred this piggyback. It might be some problem. And also, you know, the in vivo CAR T instead of ex vivo making CAR T might be the very good option. And recently, a clinical trial for CD19 already open, and wait the result. And the other way is the point of care. Everybody know that, and we can reduce the cell chain cost. And the other might be the CAR-NK and third-party donor, as you heard yesterday. There's many methods of gene editing with the TALEN or CRISPR with DNA, some repaired with the RNA. There's many ways are ongoing clinical trial, and we heard the beautiful data of NK yesterday. by Anderson group. And as noted, when you think about the Kymriah, which was historically very important, they performed only 2 in first trial, Emily, and the other one relapsed as a CD19 negative, but they success in 1. So they continue to open the some IIT in 30 patients, and then transfer to Novartis and perform SIT. Same for Yescarta and Brin. There's many clinical trials around the world, but not in Korea. So I think as a human validation is necessary for CAR T to make a history, and because there's many technical now ongoing to develop, and to develop new cell and gene therapy, small scale IIT and SIT, then SIT, then commercialization might be the best sequence. We discussed about the commercialization yesterday, but for the maker history, I think IIT is very important because we need new things. And as IIT under relatively favorable regulation and fast and optimal way to apply new treatment to human, and relatively little economic damage even after the failure, it might be the best way. But not in Korea, there's not so much IIT. So my CD19 study is a public one. First purpose is to treat the sick child, and second purpose is to make IIT system in our hospital, like US or like Europe. And we hope to validate this novel idea of a CAR T in many kind of disease in near future, and many novel CAR T or many novel cell and gene therapy might be appear in Korea in near future. I think when I explain the Kymriah, I think it's some Motorola, the first phone, only connect and very expensive. But you have Apple or have a Samsung phones in your hand, very not so expensive and very talented. So in the future, I think this kind of work might be occurred in this CAR T field, and we hope Korea also have some work in this field. I thank to my team, and thank you for your attention. Yeah.
Thank you, Professor Kang, for the insightful presentation. And now we can open the floor for questions from the audience. I have one. So what further advancements in the treatment for AL do you think are really promising, and then how that could shape the future of your research?
Somebody always asks me, which is better, Kymriah or bispecific antibody? Currently, there is no answer. Do you like father? Do you like mother? It's same. But in future, I don't know, because there is some development in the antibody field, but there's some limitation because it has no logics. But in the CAR T, we can give the logic to the cells, so better. I think that might be there is some movies that when nanobot treat every disease. Before then, I think this cell and gene therapy might be the best way to treat the patient.
Thank you. Are there any further questions? Maybe you can.
Okay, thanks. I just like to ask a question regarding the CD19/22, so like double targeting. Can you elaborate a bit more which type of indication that would work having this double targeting?
I think that might be the next way. If it might be proved after first in the patient who failed in 19 treatment, like the Berlin or like the Kymriah. After failure, we can treat the 19 and 20, and then we can go forward and maybe substitute Kymriah and others in the way.